This extended guide will walk you through the fundamental concepts of nucleic acid structure and organization, essential for understanding molecular biology. We will cover everything from the central dogma to the intricate packaging of DNA within the cell, providing detailed explanations and answering key questions. English section. Learning objectives. By the end of this tutorial, you should be able to explain information related to nucleotide structure and nomenclature. Use knowledge of the organization of DNA versus RNA. Understand general features of a chromosome. Central dogma of molecular biology. Life as we know it relies on a sophisticated system for managing genetic information. An organism must possess the capability to store and preserve its genetic blueprint, transmit this vital information to subsequent generations, and effectively express it to carry out all the complex processes necessary for life. The foundational framework that describes how genetic information flows within a biological system is known as the central dogma of molecular biology. This dogma illustrates the major steps involved in handling genetic information. Genetic information is securely stored within the precise base sequence of deoxyribonucleic acid molecules. This DNA acts as the master blueprint. From this blueprint, information can be copied through replication to create new DNA molecules, ensuring genetic continuity. Information can also be transcribed from DNA into RNA through transcription. If this RNA is messenger RNA, its information is then translated into protein through translation. In some specific cases, information can flow in reverse, from RNA back to DNA, a process known as reverse transcription. Ultimately, this stored information is retrieved and utilized during a process called gene expression to synthesize all the proteins an organism needs to function. Traditionally, a gene is defined as a specific unit of DNA that carries the instructions for encoding a particular protein or a specific type of RNA molecule. While our understanding of gene expression has become more nuanced and complex, recognizing various regulatory mechanisms and alternative splicing, this working definition remains incredibly useful for foundational understanding. Gene expression and DNA replication, a comparative overview. It is crucial to distinguish between gene expression and DNA replication, although both involve DNA. These two fundamental processes serve different biological purposes and have distinct characteristics. Gene expression is the process by which the information encoded in a gene is used to synthesize a functional gene product, typically a protein or an RNA molecule. It involves two main stages. 1. Transcription. This is the first stage, where the genetic information from a specific, relatively small section of a double-stranded DNA molecule is transferred to the base sequence of a single-stranded RNA molecule. For instance, an average human gene might be 10 to the power of 4 to 10 to the power of 5 nucleotide pairs long. In eukaryotic cells, transcription primarily takes place in the nucleus and occurs throughout the interphase. 2. Translation. If the RNA molecule produced during transcription is a messenger RNA, then translation is the process that converts the information embedded in the RNA base sequence into the specific amino acid sequence of a protein. This protein synthesis primarily occurs in the cytoplasm and is also active throughout the cell cycle. The ultimate outcome of gene expression is the production of all the diverse proteins an organism requires for its structure, function, and regulation. In contrast, DNA replication is the process by which an entire chromosome is duplicated. Its primary purpose is to ensure that when cells divide, each daughter cell receives a complete and accurate copy of the genetic information. This means that before a cell divides, every chromosome must be precisely copied. DNA replication is a highly regulated event that occurs specifically during the S phase of the cell cycle, and it takes place within the nucleus. The outcome of replication is the duplication of chromosomes, not the production of proteins. To summarize the key differences, gene expression produces all the proteins an organism needs and involves transcribing small sections of DNA into RNA in the nucleus, followed by translation in the cytoplasm throughout interphase. DNA replication duplicates entire chromosomes in the nucleus during the S phase, preparing the cell for division. The eukaryotic cell cycle. The cell cycle provides a temporal framework for understanding when these critical events occur within a eukaryotic cell. It describes the ordered series of events that take place in a cell leading to its division and duplication. The cell cycle is broadly divided into two main phases. M phase, this is the period during which the cell undergoes nuclear division and cytoplasmic division to form two genetically identical daughter cells. Interphase, this describes the period between two successive cell divisions or mitoses. Gene expression is active throughout all stages of interphase, as the cell carries out its normal functions and prepares for division. Interphase itself is further subdivided into three distinct stages. G1 phase, this is a period of significant cellular growth and normal metabolic activity that precedes DNA synthesis. During this phase, the cell synthesizes various enzymes and proteins necessary for DNA replication. Cells that have permanently exited the cell cycle and are no longer dividing, such as mature muscle cells and nerve cells, are said to be in a specialized quiescent state known as G0 phase. These cells are metabolically active but no longer proliferating. S phase, this is the critical period during which DNA replication occurs. By the end of the S phase, the cell's DNA content has effectively doubled, and each chromosome now consists of two identical copies, called sister chromatids, which are linked together at a constricted region known as the centromere. G2 phase, this is another period of cellular growth and preparation that follows DNA synthesis but precedes mitosis. During G2, the cell synthesizes proteins required for cell division, and importantly, the replicated DNA is meticulously checked for any errors or damage before the cell commits to division. The overall progression of the eukaryotic cell cycle can be visualized as a circle, starting from G1, moving to S, then G2, followed by M phase, and then back to G1 or into G0. Supplementary point: chemotherapeutic agents and the cell cycle. Many chemotherapeutic agents, which are drugs used to treat cancer, exert their effects by specifically targeting different phases of the cell cycle. This is a frequently tested concept in medical examinations because disrupting cell cycle progression can effectively inhibit the rapid proliferation of cancer cells. For instance, agents like methotrexate, 5-fluorouracil, and hydroxyurea primarily target the S phase, interfering with DNA synthesis by blocking nucleotide precursors or DNA polymerase activity. Bleomycin is known to act during the G2 phase, affecting DNA integrity and repair mechanisms, leading to DNA strand breaks. Paclitaxel, vincristine, and vinblastine target the M phase, disrupting microtubule formation and function, thus hindering chromosome segregation and cell division. Some agents, such as cyclophosphamide and cisplatin, are considered non-cell cycle specific, meaning they can affect cells in any phase of the cell cycle, often by damaging DNA directly through alkylation or cross-linking. The intricate control of the cell cycle is achieved at specific checkpoints located between the various phases. These checkpoints are regulated by strategic proteins, most notably cyclins and cyclin-dependent kinases. Cyclins are regulatory proteins whose levels fluctuate during the cell cycle, while CDKs are enzymes that, when activated by cyclins, phosphorylate target proteins to drive cell cycle progression. These molecular safeguards ensure that a cell will not progress into the next phase of the cycle until all the molecular events and conditions in the preceding phase have been successfully completed and verified. Preventing errors like incomplete DNA replication or chromosome segregation. Reverse transcription. While the central dogma traditionally describes information flow from DNA to RNA to protein, there's an important exception: reverse transcription. This process involves the synthesis of DNA copies from an RNA template. Reverse transcription is most commonly associated with the life cycles of retroviruses, such as HIV. These viruses possess RNA genomes and replicate and express their genetic information through a DNA intermediate, which is integrated into the host cell's genome as a provirus. This allows the viral genetic information to be passed on to daughter cells along with the host's own DNA. Although less prevalent, reverse transcription also occurs to a limited extent in human cells, where it plays a role in amplifying certain highly repetitive DNA sequences within the genome, such as line elements, which are a type of retrotransposon. Nucleotide structure and nomenclature. The foundational building blocks of nucleic acids, both DNA and RNA, are nucleotides. Each nucleotide is a complex molecule composed of three distinct components covalently linked together. 1. A nitrogenous base. 2. A 5-carbon sugar. 3. One or more phosphate groups. 5-carbon sugars. The type of 5-carbon sugar present determines whether the nucleic acid is RNA or DNA, and also influences the classification of nucleosides and nucleotides. The carbons of the sugar are numbered 1 foot through 5. If the pentose sugar is ribose, the nucleic acid is RNA. Ribose has a hydroxyl group at both the 2-foot and 3-carbon positions. If the pentose sugar is deoxyribose, the nucleic acid is DNA. Deoxyribose lacks a hydroxyl group at the 2-foot carbon position, having only a hydrogen atom there, hence the deoxy prefix, meaning without oxygen. This difference in the 2-foot carbon is critical for the stability and function of DNA versus RNA. Bases. There are two primary types of nitrogen-containing bases commonly found in nucleotides, distinguished by their chemical structure and the number of rings they possess: purines and pyrimidines. These bases are heterocyclic aromatic compounds. Purines: these bases are characterized by a double ring structure, consisting of a 6-membered ring fused to a 5-membered ring. The two purines commonly found in both DNA and RNA are adenine and guanine. They are essential for forming the genetic code. It's worth noting that other purine metabolites, such as xanthine, hypoxanthine, and uric acid, exist in biological systems as intermediates in purine metabolism but are not typically incorporated into nucleic acids. Pyrimidines: these bases have a single ring structure, specifically a 6-membered ring. Cytosine is a pyrimidine found in both DNA and RNA. Thymine is almost exclusively found in DNA, while uracil is typically found only in RNA, where it replaces thymine. The presence of a methyl group on thymine distinguishes it from uracil. Nucleosides and nucleotides. The assembly of these components follows a specific order. Nucleosides are formed when a nitrogenous base is covalently linked to the number 1 carbon of a 5-carbon sugar via a beta-N glycosidic bond. To differentiate the carbons of the sugar from the carbons within the purine or pyrimidine base, the sugar carbons are labeled with primes. For example, when adenine is linked to ribose, it forms the nucleoside adenosine. If adenine is linked to deoxyribose, it forms deoxyadenosine. Similarly, guanine linked to deoxyribose forms deoxyguanosine, and uracil linked to ribose forms uridine. Nucleotides are then formed when one or more phosphate groups are attached to the 5' carbon of a nucleoside, forming a phosphoester bond. For instance, if one phosphate group is attached to adenosine, it becomes adenosine monophosphate. If 2 phosphates are attached, it's adenosine diphosphate, and with 3 phosphates, it's adenosine triphosphate. Nucleoside D-N triphosphates, such as ATP, GTP, CTP, UTP, and their deoxyribose counterparts, are particularly important because they contain high-energy bonds. The hydrolysis of these bonds releases a significant amount of energy, making them crucial for cellular energy transfer, signaling, and as precursors for nucleic acid synthesis. For example, ATP has 2 high-energy phosphate bonds. Let's clarify the nomenclature for the important bases, nucleosides, and nucleotides, for the base adenine. Ribonucleoside, adenosine. Ribonucleotides, AMP, ADP, ATP. Deoxyribonucleoside, deoxyadenosine. Deoxyribonucleotides, DAMP, DADP, DATP. For the base guanine. Ribonucleoside, guanosine. Ribonucleotides, GMP, GDP, GTP. Deoxyribonucleoside, deoxyguanosine. Deoxyribonucleotides, DGMP, DGDP, DGTP. For the base cytosine. Ribonucleoside, cytidine. Ribonucleotides, CMP, CDP, CTP. Deoxyribonucleoside, deoxycytidine. Deoxyribonucleotides, DCMP, DCDP, DCTP. For the base uracil. Ribonucleoside, uridine. Ribonucleotides, UMP, UDP, UTP. Deoxyribonucleoside, deoxyuridine. Deoxyribonucleotides, DUMP, DUDP, DUTP. For the base thymine. Ribonucleoside. Ribonucleotides. Deoxyribonucleoside, deoxythymidine. Deoxyribonucleotides, DTMP, DTDP, DTTP.